Gastrulation, that is, if you remember that we had done blastulation in the last class. After blastulation, we are going to talk about gastrulation. So what exactly is gastrulation is we are going to talk about in detail in this particular class that we are going to have. It's going to be again interesting class over here. Now, coming to gastrulation, so but before going into gastrulation, I would like to talk about this point particularly, where I want to have a recap for you all. That is, we had talked about fertilization, and after fertilization, a zygote is formed. The once zygote is formed, it has to undergo cleavage. Cleavage occurs because of mitosis. Here, the mitosis over here, how it occurs is it is not going to grow in size. Whatever the size of the zygote is, the cleavage is going to be of the same size here. It's only the cytoplasm that is going to get distributed equally among all the cells, and there is no growth in the size as such. Okay, so here cleavage, that is depending on different types of species. Over here we talked about different types of cleavages. If you remember, we talked about spiral cleavage, radial cleavage, rotational cleavage, and also we talked about types of eggs where the amount of yolk is going to decide whether it is going to be microlecital, alecital, mesolecital, and megalecital. So once the egg undergoes cleavage over here, the zygote undergoes cleavage. It has to go through 2 cell, 4 cell, 8 cell, so on, and then it goes into a morula stage. And then morula stage, when the coelom is present, blastocyst is present over here. That is called as a blastula stage over here. Now, once a blastula is formed, it has to undergo a gastrulation phase. What do you mean by a gastrulation phase? In Amphioxus, gastrulation means the formation of gut over there, and also the production of the three different germ layers that you're going to talk about over here. That is the ectoderm, mesoderm, endoderm. Now, once the zygote undergoes cleavage, it has to go to blastula stage. From the blastula, it has to go to gastrulation stage. What exactly is gastrulation? We are going to talk about the formation of the three germ layers, that is the ectoderm, mesoderm, endoderm, which is going to be the future organs of any species that it is going to form. So we are going to talk about this stage in this particular class over here, and also organogenesis will be dealt in next class. Okay, now coming to what exactly is gastrulation, we'll talk a little bit about the meanings of this gastrulation over here. The term gastrulation means, that is, it is from the Greek word belly formation. So what exactly is a belly formation? That is a digestive tract formation is what you're going to talk about as gastrulation. But when you're talking about it in a broad spectrum, as I told you, in Amphioxus, the formation of GI tract is because of this archenteron that is being formed, and the process of this archenteron formation is called as gastrulation. Gastrula means belly, and formation of a GI tract is what is called as gastrulation. But as I said, when you're going to talk about in a broad spectrum, that is when you're going to talk about the higher organisms, during gastrulation, what happens is the three germ layers formation is going to occur. What are the three germ layers? That is the ectoderm, mesoderm, and endoderm. So although there is a difference over here, but what scientists have decided it is they will go according to this name gastrulation itself, because otherwise there will be a confusion. So gastrulation stays for all the different organisms as well. Then this another great scientist, Hans Spearman, he was the one who identified this in amphibians, and so it is also called as Spearman's organizer, where the Spearman's organizer is where the dent is going to be formed, from where the involution of the cells occurs to form these three germ layers. So that is called as Spearman's organizer by Hans Spearman. Okay, so the same region in the birds is also known as Hense's node. If you remember, when you were talking about this in the practical class, we were talking about something called primitive streak. The primitive streak also has a primitive node, a primitive pit over there. So at that place over there, it's called as Hense's node. How? Victor Hensen was the one who identified that, and so the name primitive node or Hense's node is proposed. Okay, so Now, coming to balance key, again, because we'll have to talk a little bit about the scientists who have really done this particular work. Otherwise, we wouldn't have known what is this gastrulation, blastulation, all those things. So, balance key, 1981, he said that gastrulation is a dynamic process in which one single layered blastula by morphogenetic movements of the cell is going to be converted into three germ layers, that is ectoderm, endoderm, mesoderm, and so it is known as gastrulation. Now I remember even one more great scientist, he said that it is not life, it is not death, it is not marriage which is important in a human's life. What is important is the zygote which undergoes cleavage and then the gastrula period. So this gastrulation is very, very, very important in one's life. Why? Because this is the one that is going to decide which cell is going to form the germ layer, and that germ layer is going to give rise to what type of organs over there. So if all this is going to be placed correctly one below the other, only then we are going to have our proper organs. If there is going to be a change in this, then definitely the organism is not going to form like what an organism has to look like. So gastrulation is very, very important in one's life. Okay, each germ layer corresponds to the development of the specific primitive systems during organogenesis. What is organogenesis? Formation of organs over here. And in addition to setting up of the embryo up for organ formation, gastrulation provides a mechanism for developing multi-level body plan that demarcates anatomical axis. So what is an anatomical axis? Now, anterior, posterior. Anterior is the face is anterior region, the tail is the posterior region, then the dorsal region, ventral region. So these are the axis of the body, and this axis is again formed because of the gastrulation over here. So if this was not there, then definitely the organism will go for a toss. You will not have a proper development of organs. And gastrulation also provides the retention of global left, right. So you're going to have your the right and the left symmetry, and also the loss of bilateral symmetry in specific organ. That is, we are going to have a left-right symmetry. That is bilateral symmetry. When you cut an organism into two, it should have two equal halves on either side, right? So that is called as bilateral symmetry. But within that bilateral symmetry, the heart of an organism over here, for example, if you're going to talk about the mammals over here, heart has lost its bilateral symmetry. So that is the reason if you cannot cut a heart into two equal halves over there. So only the heart is the one that has lost its bilateral symmetry. So the gastrulation is going to decide which is going to be bilateral symmetry, which is going to lose its bilateral symmetry, why it is going to lose its bilateral symmetry. Everything is set up in the cell, and each cell is going to get the information. That is during this gastrulation, the starting of the information from the DNA is the transcription is going to start over there, where each and every signal is given to start what should precede what. Okay, so here when you're going to talk about this embryonic disc over there, this is the disc that you're going to see it under on the egg. For example, if you're going to talk about the chicken over here, so you're going to see this is as a primitive streak over here, and that's the primitive pit or a hensis node over there. And the layers that is on top over here, these are the epiblast over there, and the cells beneath them. So here it is, this is not a 3D picture that I can show you one below the other. So below that, this is the area beside this epiblast is the ectoderm, and area below is a hypoblast, which will be showing over here. So if I'm going to cut this into half like this and going to show them in the transverse section over here. So this part, if you're going to cut them in the transverse section over here, so that's then amniotic cavity over there, and that's a yolk sac over there. So this is a mammalian zygote that is I'm showing you all over here after the division, and this is a gastrulation that has occurred over here. Now this amniotic cavity over there, inside this cavity is the one that is going to take care of the developing embryo over here. So that's the formation of the embryo over there, and that's an ectoderm over there that is going to be formed. The epiblast, which is going to give rise to new cells over there, that's the mesoderm in underneath, and in between these, and after the ectoderm and the mesoderm, the one that is going to be below, that's the endoderm. So future endoderm is what we are going to see over here. And of course, you have a yolk stalk over there. This is going to form what you're going to be forming. We'll talk about that in a later class. So here, this is a secondary, that's a yolk sac over there, where a little bit of yolk that is present in the mammal. So you'll be saying that mammals don't have yolk. Yes, they are alecytic, but the little amount that is going to be present, that is the one that is going to nourish the development of the future gastrulation period of that, not completely, only for a few maybe few days over there, until this yolk sac over here, the hypoblast over there, that is going to form the placental formation that is going to be formed by the syncytioblast over there and cytoblast. We'll be talking about that syncytio and cytoblast. So what I want you all to understand over here is we are going to talk about how gastrulation occurs. Gastrulation occurs via 5 different types over here. So what is this 5 different types of here? Movements of the cells that is going to move in different patterns over there. So what is this pattern over here? Invagination, involution, ingression, delamination, and epiboly, and emboli also is there. We'll talk about emboli when you're talking about that point over there. So what is this invagination, involution, ingression, delamination, epiboly? These are a way in which the cell is going to move from one place to another place and occupy that place permanently. So when you're talking about invagination, so for example, imagine you have a rubber ball, and if you place a dent over there, you're going to have a dent. Now from that dent, the cell moves inward. So now if you can see in this picture over here, example of a sea urchin over here. So there's a dent over there, and if you make a dent over there, that it is going to go inside, infolding of the cell is going to occur. Now that is called as invagination. Involution is, there's a difference over there in invagination and involution. There is a dent over here also, but what is happening is the cells on either side, if you can see the cells on the either side over here, now that's a blastopore, the cell is going to involute and they are going to bend and going to take a round shape over there, right? So they're going to take a U-turn over there and they're going to go around over there. So that's a U-turn and going to go around over here. That is called as involution. Now coming to ingression, ingression is the cells that are present on the hypoblast, that is the lower side of the cell, the blastula stage over there. Now this cells is going to give rise to some new cells over there, and these are the one that is going to migrate individually into the embryo. Now that is called as ingression over there. Delamination is the cells that are going to be present, that is going to give rise to new cells, but the cells are not going to be allowed to go inside the embryo. They're going to get attached to the layer above it. And after sometimes when the cells are going to be formed, this will split and they're going to form a new sheet over there. So that is called as delamination. Epiboly is extension of the outer layer over there. So for example, now when something like involution occurs over here, the involution occurs, the expansion of this ectodermal cells goes around the embryo, and then they come over there near the blastopore and closes them over there. So that is called as epiboly. Covering the whole embryo is called as epiboly over there. So these are the patterns. Now you'll have to understand when I'm saying invagination, you'll have to remember when I'm saying involution of the cells, you'll have to remember how they take a U-turn, and ingression, how they separate, delamination, how the two cells are going to be formed, epiboly, how the cells are going to cover the embryo completely. So these are the pattern in which the cells are going to move from one place to another place over there. Now, what is the characters of gastrulation? You'll have to understand the characters first. So when you're talking about the characters, so this is the first stage of cellular differentiation. As I told you, gastrulation is a very, very important stage. And this is the first stage of cellular differentiation. An embryonic cell begins to follow in a specific layers through morphogenetic movement. morphogenetic movement. Everything is genetics over there. And the formation of three embryonic layers, that is the ectoderm, endoderm, and the mesoderm. So these are going to be the main characters over here. And this cellular division is synchronized between them. So synchronization of the ectoderm, endoderm, mesoderm formation is all synchronized. It's not something like, okay, first ectoderm is going to form, then the mesoderm, then the endoderm. No, everything is going to be formed together. So that's a synchronization. And metabolism changes within the cell due to oxidative process becomes dominant, because if they have to divide, they need some energy. Metabolism also has to occur over there. And the nucleus at this stage has more impact than before. That is in the process of controlling of the development. Till the bachelor stage, the nucleus is not activated. When it comes to gastrulation, the nucleus is activated. Why the nucleus is going to be activated? Because the DNA has the information of that particular cell, the signals that is going to be produced at that particular time. And this signals, when activated, will give the next step of what the cells has to perform, right? So that is, nucleus is at this stage has more impact over there. And here it shows that the chromosomal, the paternal genes and its effects has a more important effect over here. Of course, paternal and maternal are equally important. Why? It all depends on what type of genes you have inherited from which parent during recombinations. Okay. Now, gastrulation rearrangement of the cells into the three-layered one is called as gastrulation, and a primitive gut is formed over here. So, for example, over here, so that is a blastocyst over there. Blastocyst is having a blastocele, and there's a dent that is going to be formed. And as the dent is going to form, now this is called as invagination. As the invagination is going to be formed, the archenteron is going to be formed, and this archenteron is the one that is going to be a future gastrointestinal or digestive tract that is going to be formed over there. Okay, now coming to what are the three layers and how they are going to be formed. So, for example, in the gastrulation over here, as we talked about, they are going to be formed into three layers. Now, that is the ectoderm, that's the mesoderm, that's the endoderm over here. The ectoderm is going to form into two parts over there, the neural plate for the formation of the brain, and that is the epidermis for the formation of the skin. The mesoderm is going to form the muscles, the kidney, the bones, and the gonads are all because of the mesodermal layer. And the endoderm is going to form the digestive glands, the lungs, and so on and so forth. Now, that is the animal pole where you have the blastoderm, and that's the vegetal pole where you have the yolk connection still over there. So that is why you're calling them as vegetative pole over there. So that's a picture over here, how you can see the ectoderm going to form the nervous system epidermis, the mesoderm going to form somites, blood, kidney, and that's the endoderm over there. Okay, now what is a derivative? So yeah, this is the same thing what we're talking about over there. Each layer going to form which part is given in the a clear picture, just one slide, and you can understand what we are going to talk about, how the mesoderm is going to form different layers, how the ectoderm is going to form the skin, neuron, and the pigments, the body pigment cells. Whether a person is going to be fair or dark or medium, it's because of this pigment of the ectoderm that is going to be formed of there, and endoderm giving rise to different GI linings and all those. Okay, so this is again, we talked about that earlier. Now coming to here, we are going to talk about gastrulation in different species. We are going to talk about amphioxus. We are going to talk about in fishes, then amphibians, aves, and mammals over there. So 5. I want you all to take a paper, the book, a pen, and then you're going to just note down a gist of it, what exactly is happening in which type of species. Now coming to amphioxus. What's an amphioxus? It's an cephalochordata. Why cephalochordata? Cephalochordata is because the notochord runs from the head region till the tail region over there. And because of the presence of the notochord still in the animals, you call them as cephalochordates, chordate over there. Okay, so what we are going to talk about in the amphioxus and how they are going to become adult. So gastrulation in amphioxus is initiated when the blastoderm, so that's the blastula stage over there, and the blastoderm at the vegetal pole. Why vegetal pole we are saying? The anterior region, that is the animal pole, the cells are a bit smaller. But when you talk about the vegetative pole over there, the cells are going to be a little bit bigger and columnar over there. Now this is going to start to become flat over there. You can see over there. So they're going to become flat over there. And once they are flat, there's a dent that is going to be formed. And because of the dent, they keep moving inside. And once they keep moving inside, they slowly form a cup shape over there. You can see this cup shape over there. Now, as the cup shape is going to be formed, now you can see the black color cells that are forming over there. Now that is the one that is going to form the future notochord of the amphioxus gastrulation stage over there. And as this cup is going to be formed, there the cells that are going to be present over there. Now this comes close by over there. As they're going to form a close by, that is, they're going to form a dent, and then they're going to close up over there. Now, as this is going to be bent, now single cell has become double cell over there. Can you see the double cell over there? Yeah. So the outside one, that is going to be the ectoderm, the between the mesoderm and the endoderm, that is going to cover the cell. And the blastocele that was present that big over there, the blastocele is no more to be found over there, because the second layer is going to completely cover them. And in place of it, an archenteron is going to be formed over there. And this archenteron is the one that is going to be forming the future GI or the digestive tract of the amphioxus over there. And this process is driven by three primary cell movement. What are they? Invagination, involution, and epiboly. Now, invagination, if you can see over there, there's a dent forming, and then invagination is occurring. Involution is, now you can see the cells, they are taking a U-turn, and they're going to move this side and this side. Now, that is called as involution. Epiboly is the ectoderm that is present on the outside is going to completely cover them, cover the whole embryo over there. How are they going to be done over here? In this picture, if you can see, invagination over here. So you have this single layered blastoderm over there at the vegetative pole over there, and this gradually folds. And as they are moving inside, the blastocele is decreasing in size over there. So you can see the blastocele that is going to be decreasing, and an archenteron is formed. And this is because of the invagination. The next one is involution over here. The cells at the dorsal and the lateral one, they're going to take a U-turn over there, and the blastopore divides rapidly and roll inwards over there. So you can see the rolling of the cells inwards to occupy the space between the outer and the inner endoderm. And as they are moving over there, the involution occurs. So the U-turn that it is going to occur, that's an involution of the cells. So here you can see the outer ectoderm and then inner endoderm over there that is going to be formed. And this involution gives rise to notochord and mesoderm. The future mesoderm is going to be formed from this layer over there. And epiboly over there. Now, when talking about the epiboly over here, now you can see the inner layer, that is the endoderm, has been formed, and the side ones, that's the mesoderm over there. The pink color ones is the mesoderm, and the green color ones is the future notochord over there that is going to be formed. And above that is the the ectodermal region that is going to be formed over there, the epiblast, that is going to give rise to neural plate over there. Now, as this is forming, there is a gap over there. Can you see that gap over there? Now, this gap is going to be filled by this ectoderm around the embryo, growing embryo, and they're going to get closed over here. Now, can you see that has been closed over there? So the ectoderm is going to completely cover the developing embryo over there, and that is called as epiboly. Now, as this is happening, you can see the archenteron being formed. The blastocele which was there earlier, that is completely gone. There is no more blastocele that is going to be seen. And in place, you have another gut formation over here, and that gut is called as the archenteron. And this blastopore is covered by the ectoderm over there by epiboly. And this archenteron is the one that is going to be forming the digestive tract. And because of the formation of this digestive tract, the name gastrulation was given over there because of the formation of the GI. Okay, so here you can see the resulting germ layers over here. What are the resulting germ layers over there? So you have this outer ectoderm, which is going to be forming the nervous system and the epidermis. The mesoderm, the in-between one. So this is the mesoderm over there. The middle layer, which is going to form the notochord. So this is going to form the notochord over there. The side ones are going to be forming the muscles and the coelom. the body cavity over there. And the endoderm, the one that is going to be in the center, the endoderm yellow color cells, these are the innermost layer lining the archenteron over there, which is going to develop into digestive tract and associated organs. So Amphioxus serves as a classical model for chordate embryology because its gastrulation is relatively simple and unhampered by the presence of the yolk. So there is nothing happening, simple gastrulation. And so this serves as a classic model for gastrulation over there. So that was Amphioxus. Now coming to gastrulation in the fish. So we'll talk about the fishes, that is a Pisces. What is happening in the Pisces over here? The first cell movement of the fish gastrulation is epibole. Complete covering is the first starting of gastrulation. Now you're going to understand over here, each species is going to have a different type of movement of the cells over here. So here, epibole is going to be the first one. In Amphioxus, we saw invagination, involution, and then epibole. Here, it is going to be the epibole that is going to be formed, that is covering up of the cells over here. So later in this, you're going to see three processes: epibole, involution, and convergent extension. Convergent extension is once the cell is formed, they're going to extend around. That is called as convergent extension over there. So we'll talk about this. So that's the epibole, covering up epibole. Involution, U-turn of the cells is the involution, and convergent. Now this place is going to increase in size and they're going to move around. That is called as convergent extension. We'll talk about that. Now coming to epibole over there. So it has been most extensively studied one in zebrafish, in the fishes, zebrafish. Now zebrafish is a very nice example for say even for toxicological point of view, zebrafish is used because they share a good genome sequence, genome comparisons with humans also. So zebrafish is used over there. So this has been studied in zebrafish as the development allows an easy visualization process shortly after MBT. What is MBT? If you remember, when I was talking about cleavage, we talked about MBT also, mid-blastular transition over there. So during this occurs in the mid-blastular transition. Margins move down progressively to cover the yolk sac of this. So you have this animal pole, you have the vegetal pole. Vegetal pole is always where the yolk is present. Animal pole is always where the blastoderm is present over there. Now, the movement of the margin around is what the cell that is going to cover by epibole to cover the yolk cells. And how much of yolk cells has been surrounded depends on the percentage of the epibole. If this is there, that's a 50%, then 90% and 100%. We're going to talk about them. And these are driven by yolk syncytial cells over there. Now, this layer is called as yolk syncytial cell nuclei. Now, this is the one, the nuclei that is present in the yolk syncytial layer. Now, this nucleus is the one that is going to give signals to the cells inside. So you can see the deep cells over there, that's a superficial epiblast over there, the formation of the ectoderm. These are the deep cells over there. These deep cells are going to be given signals to either move or which way they have to move, which way they have to involute. Everything is given by this yolk syncytial nucleus over there. And then what's happening? Formation of hypoblast either by involution. So if you can see over there, either by involution, the hypoblast is being formed, or the epibole blastoderm by delamination can also occur. That is after 6 hours of formation over there. So here you can see you have the outer layer over there, then the epiblast that is being present out, that is ectoderm, the epiblast over there. Then you have an hypoblast formation, that is involution is occurring over here, and you have a shield over here. Now, what is the shield over there? There's a particular name for the shield. The shield over here is called as a germ ring or shield, you can call them as, or you can call them as embryonic shield over there. So involution occurs after 50% of epibole over there. So what is epibole over there? Here you have found the movement of the cells around the yolk is epibole. And at a place called as a germ ring, involution occurs. Because again, they have to have a signal where they have to involute, right? So involution occurs at a germ region over there. And this cells, when they start to involute, they are going to form the mesoderm or the mesodermal layer, or it's also called as epiblast, hypoblast, because it is below the epiblast. Now, I want you to again remember, epiblast is the upper layer, hypoblast is a lower layer. So whenever we say epiblast and hypoblast, we're going to understand that the outer layer is the epiblast, and the one that is below the epiblast is called hypo. Hypo is below. So the name hypoblast over there. So at 50% epiboly, formation of hypoblast or the mesodermal layer occurs, and the movement of the mesodermal cells takes place from the germ ring over there. And this is carried out by the deep layer cells over there, the epiblast layers that is deep inside. So these are the one that is going to form this hypoblast or the mesoderm. And where it is going to be forming from, it's going to be forming from the germ ring over there, or embryonic shield over there. So you can see over here how the epiboly is occurring over there. And as the epiboly is occurring, the germ shield over there, from where involution is occurring. And as the involution is occurring, then the next stage we are going to talk about is the convergence stage. So before that, we'll see a little bit of this point over there when you're magnifying this point over there. So you can find that's the enveloping layer of there. Below that enveloping layer, you have this epiblast. The epiblast is going to give rise to cells that is going to form the involution. These are the cells that is not going to involute, but this is the one that is going to direct them to go inside over there. That is the one that is going to form the hypoblast over there. And these are the syncytial yolk layer, where the nucleus is very much active, and this is the one that is going to give signals to the deep layers to form this involutions over there. Okay, at 90% of epiboly, now after 9 hours, so you can see the epiboly is slowly covering complete the zygote is being completely covered over here. So if you, here you can see it is only 50%, and here you can see completely it has covered. And the mesoderm can be seen surrounding the yolk over there, and the inside one is the endoderm that is going to be formed over there. And at 10 hours, completion of gastrulation occurs over there. So you have the animal pole, a broad region. So that's going to be the head region over there. This place where the mesoderm is going to be formed, that is the place that is going to give rise to somites. The somites, if you remember, I told you all, somite is going to give rise to the muscles and other connective tissues over there. And then you have this the tail region over there, the tail bud that is going to be formed at the vegetal pole over there. So these are the different parts that is going to be formed after 10 hours of gastrulation. Now, once this is going to be formed, there's also, as this is going to be formed, there's also one more thing that is going to occur, that is a convergent extension. What is this convergent extension? The dorsal line elongates, becoming the midline over there. So you have this midline of the anterior posterior body axis, anterior and posterior body axis. That's the anterior region, and that's a posterior region over there. Or if you can see in this particular graph over here, that's the anterior and that's a posterior region over there. So a dorsal line elongates in the mid region, forming the anterior posterior body axis, and that is the ventral and the dorsal region. The dorsal region, ventral region, anterior, posterior. And if I'm going to keep my hand like this, anterior, posterior, and dorsal and ventral region over there. The lateral tissue converges in the midline and elongates, and then the mid-dorsal, the mid-ventral that is going to be formed does not participate in the dorsal convergent. Why? Because this is the part where it is going to be the tail region. So there is no convergence over here. Most of the convergent extension is going to be formed in the anterior posterior region. How this is going to be formed, you can see over here. Now, if you can see the convergent extension, that's a mid-dorsal line. The extension is going to go up like that. So if you can see, it has gone up like that. Convergent extension is happening over there. As this is happening, it becomes even more narrower. And as this is narrowing, you can find that this has become something like that over there. That is the head region, and that is a tail region. That is the tail bud region. That is, sorry, that is going to be formed over there. And the convergent extension of epiblast forms the neural keel over there, or the neural tube that is going to be formed. Okay, coming to gastrulation of frogs. So that was about fishes, how they are going to be formed. So you have the head region, that is the brain that is going to be formed over there. You have the somites that is going to be formed, and that is the tail region that is going to be formed. So that is gastrulation in fishes. Now coming to gastrulation of the frog, that is the amphibians. Vegetal rotation and invagination of the bottle cells. There is something, the cells are going to become bottle cells. That is, what is bottle cells over there? The epiblast is the one that is going to take the shape of a bottle. So it is going to narrow at one place, broader at the other place. That's why it's called as bottle cells. And this bottle cells, what they do is they invaginate inside. Involution at the blastopore lip, convergent extension of the dorsal mesoderm region, and epiboly of the prospective ectoderm occurs. Epiboly, that is covering up by the ectoderm, is going to occur. So coming to what gastrulation is all about in the frog over here. Gastrulation begins when the cells on the dorsal side invaginate, cells on the dorsal side. So that's a dorsal side. They're going to invaginate to form a small dent or crease at the blastopore layer. So here again, you'll have to understand now, after the blastula stage, when it is going to come to gastrulation stage, then you can see at one particular place, this is going to occur. It's not going to occur at any random place, the dent that is going to take place. So it is definitely going to take place only at the dorsal lip of the blastopore. You can see a small dent has been formed over here. And as the dent is formed, the cells from the animal pole is going to come around and they're going to invaginate inside over there. So they're going to invaginate over there. So you can see the cells over here that is going to invaginate. A sheet of cells begins to spread out of the animal hemisphere, rolls inside over the dorsal lip, that is for the involution, and moves into the interior of the cell over there. So you're going to see how they are going to move inside. So can you see the movement over here? The red color one has moved inside over there. So that is the involution over there. In the interior, these cells will form the endoderm and the mesoderm. So these cells is going to start to form endoderm and the mesoderm with the endodermal layer, of course, on the inner side. Meanwhile, the cells at the animal pole changes shape and begins to spread around or over the sheet over there. And the blastopore around both sides of the embryo are more or less going to invaginate. And when the ends are going to meet, the blastopore forms a circle. They are going to form a small circle over there. Can you see the small circle over here? Now, this small circle is going to be called as a yolk plug. Why it is called as a yolk plug is because this plug is the one that is going to be in contact with the yolk of uh, the egg over there. So that is going to be a yolk plug where nutrition has to come for the development of the frog, because here, if you understand or remember, the development of the embryo, the mother, it is not going to give nourishment. The mother is not going to give nourishment. Why? Because during the development of the egg, all the yolk is being given to one particular egg, and maximum amount of cytoplasm and yolk is collected in the egg. And during the embryo development, the yolk is the one that is going to nourish the development of the embryo over there. So here, that is the yolk plug over there. Although the embryo, ectoderm is going to cover by epiboly, so you have a one small part over there that is called as a yolk plug. So it's not going to be completely covered up over there. So internally continued involution expands and the endoderm and the mesoderm, and there's an archenteron that is being formed. So that's an archenteron. And as the archenteron is being formed, the blastocele is shrinking over there. You can see. So that was the biggest blastocele over there. And this blastocele is going to shrink in size over there. And this shrinking of the size is going to lessen up, and completely you have an archenteron that is going to be formed over there. Okay, so late in the gastrulation, the cells remaining on the surface make up the endoderm. So the cells, the outside is going to make the ectoderm. The endoderm is the innermost layer, and the mesoderm lies in the in between the ectoderm and the endoderm. Sorry, I'm having some problem with it. Okay. So you have this stages over there, the ectoderm, the mesoderm, and the endoderm, and the endoderm is going to line the archenteron. So what is the fate maps of the germ layers over here? The ectoderm is going to form the epidermis, cutaneous glands, eyes, cornea, retina, whereas the mesoderm is going to form the notochord, pericardium, muscles, skeleton, connective tissues. The blood is most important connective tissue over there. And endoderm is going to form the digestive tract, the respiratory tract, the eustachian tube. What is the eustachian tube? That is the ear tube that is going to give us the balance of a human. If you're able to balance, that is because of the eustachian tube. Ever imagine when somebody is going to bang us onto the ear, and it goes for a toss and you lose the balance over there. So eustachian tubes is given by the endoderm over there. And of course, the pancreas, the pancreatic ducts, the urinary bladder is all derived from this particular endoderm layer. So that was about the frog. Now coming to gastrulation in chicks. Now I'm going to pause for a while and I'm asking y'all, what is the difference between these two? So if I'm going to ask you all, what is the difference between these two? Now that is, both are from the chicken. Okay, so we are going to talk about the gastrulation in the chick. Both are from the chicken. And this is one type of an egg, and that's one type of an egg. So I suppose most of y'all must have made omelette at home, right? Now, when you break open an egg, and when you notice there's a small white dot over there, can you see that small white dot over there? There's this white dot over here also. Now, how you differentiate between a fertilized and an unfertilized egg over here? So this small dot, white color dot over here is a fertilized egg, and that's an this is an unfertilized egg, and that's a fertilized egg. So you can see that a small transparent layer in the center, and then there's a disc of area opaca over there, and that's the area pellucida over there. Now that's a fertilized egg and an unfertilized egg. You must be noticing how do we find out? If you are living in some villages where you have your small poultry at home, definitely whatever the eggs that you all are eating is a fertilized eggs. But the eggs that we buy from outside, the broiler eggs that we buy from the smart shops and all those over there. So this is unfertilized egg because these eggs that are laid by the hen is unfertilized over there. So that's the difference between fertilized and unfertilized eggs over there. So we are going to talk about the chicken gastrulation. Gastrulation in chick is prolonged due to the large amount of yolk. Why? Because the size of the chicken is a little bit bigger one to grow. So what is the incubation time for the egg? 21 days. This 21 days, the embryo has to depend on the yolk that is given by its mother when the egg is going to be formed in the isthmus of the chicken, female hen. Now they have to take 21 days, and 21 days the egg has to have food inside. And that is the reason why the hen gives rise to large amount of yolk, and that is called as mesolecital or megalecital. Mega means more, meso is medium, mega is more, megalecital egg. So amount of yolk is more in them. So it starts about 4 to 5. What gastrulation starts about 4 to 5 hours after laying and completes by second day of incubation. Now, blastula is as the egg has been formed, blastulation has occurred. Now, as I told you again, if it is a country hen, then definitely fertilization occurs when the sperms are inside, and ovulation occurs, the egg is going to get fertilized over there. Now, once this starts, now blastulation, after the laying of the egg, it's up till blastulation. And then the next stage of development, that is a gastrulation stage, starts only when the female hen sits on the eggs and incubate them. Only then you will find the gastrulation stage. So if the hen is not going to incubate, then definitely there is no more stages that is going to be formed, while the temperature is necessary for the development of further stages of the embryo. So at the onset of the gastrulation in chicken, upper layer and the lower layer. So this is the layer that is being the blastocyst layer that is going to be formed. Now gastrulation stage when they occur, so you can find the separation of the cell is going to occur, and they're going to give rise to epiblast and the hypoblast layer. So two layers, epi, always remember epi on top, hypo below there. Epi and the hypoblast is going to start to form over there. And following the gastrulation, two types of morphogenetic movements occur. Emboli and epiboli. Here, you're going to talk about one more type of new terminology, emboli. Till now, we talked about invagination, involution, ingression, delamination, and epiboli. Now, we're going to talk about emboli. Now, what is the emboli? It involves only the epiblast, which contains cells of ectoderm, mesoderm, and notochordal cells. It includes convergence, invagination, and involution, and a formation of primitive streak that is going to occur over there. That's called as emboli. What is this emboli? Epiboli is covering of the embryo completely, is epiboli, outer side. Emboli is when a primitive streak, so for example, if I'm going to talk about this primitive streak over there, when the primitive streak is formed, the cells from the epiblast is going to go inside. And as they are going to move from all the side, now this is called as emboli. So that's a difference over there. And this is going to give rise to the notochordal cells, the mesoderm cells, and the endoderm. That is the hypoblast cells, which is going to form into an endoderm in the later stage. Now I want you all to understand one more thing is the development of gastrulation in the birds is same as what you're going to talk about in the mammals also. So what I want you all to understand is when I'm talking about this chicken gastrulation, if you understand this, then the mammal gastrulation is simple over there. So posterior margin of the blastoderm contains a local thickening called as Kohler's sickle. That thickening over there at one particular place, that's a posterior side, is called as a Kohler's sickle over there. Something like the germinal ring that we saw in the fishes over there. So something like that. You have a Kohler's sickle over there. Now this Kohler's sickle, that is a thickening of the cells over there, is a local thickening of the epiblast. So you have this epiblast. There's a local thickening over there at the posterior edge of area pellucida. Now, I want you all to understand what is area pellucida, area opaca. During the formation of the egg, area pellucida is where the embryo is going to be formed. Area opaca is around them, where the blood islands is going to be formed over there. So it acts as a critical development organizer, something like the Spearman's organizer, driving gastrulation by initiating the formation of primitive streak. So you're going to start with a primitive streak over there at the posterior region where the Kohler's sickle is going to be formed over there. So here you can see that's the area pellucida, that is the epiblast that is going to be formed, and around them that is the area opaca, that's the area opaca over there. And as the cells are moving inside, the cells are moving inside by process called delamination of epiblast. So that's a cell over there. Delamination is the cells from the epiblast moves, they get cut off over there, connection gets cut off, and these cells move inside the cells, and that is called as delamination of the epiblast into the cavity of the epiblast cells over there, which is going to be formed slowly into hypoblast and the mesodermal cells over there. So as soon as the delamination of the hypoblast cells starts to form a sheet of cells from the Kohler region. So from the Kohler region, you can find how the sheets are going to be migrating to the anterior side and joining the invagination islands, thus forming a secondary hypoblast. How the secondary hypoblast is going to be formed over there, we'll be talking about this. So as the movement of the cell is going to occur, so that is the Kohler's sickle over there. As the cells are going to be coming inside, the Kohler's sickle is going to elongate. So here how the cells are going to be moving towards the anterior region. So this is the anterior part, that's a posterior part. Kohler's sickle is at a posterior part. The cells keep moving in over there. As the cells are going to move over there, slowly there is a primitive streak that is going to be formed over here. So that is a primitive streak. The Hence's node is going to be formed. The primitive, that's the area pellucida, that's the area opaca over there. And you have the primitive groove over there. What's a primitive groove? The groove is a place. So if you can see the groove over there, this is a place where the epiblast is going to converge inside, and they're going to go inside by a process called emboli. I told you emboli, the movement over there. So here you have the primitive streak over there. The primitive streak is going to take a complete midline, that is almost 80% of the area pellucida is taken over by the primitive streak over here. So this is the primitive streak that is being formed. That's a Kohler's sickle that is present over there. And as the primitive streak is formed, what happens over there is the next step that we are going to talk about over there. Now, once the primitive streak is formed, this will tell the cells from the epiblast. Now you can see that's the epiblast over there. So this is a single layer epiblast. So this is the cells of the epiblast. And what's happening is the cells from either side, they start to move inside this primitive streak. The primitive streak is going to form a groove over there. Now this groove is the place where the cell is going to start to slowly come inside. As they are going to start to come inside, just give me a break. With last night food over there. Okay, so yeah, so what's happening is there is a primitive streak that is going to be formed. This primitive streak is going to form slowly a primitive groove over there. Now because the groove are present, the epiblast that are present over here, these are going to move inside this groove over there. As they are moving inside the groove, so you can see a space over there. Inside this, the cells are going to come inside. Now what is happening over here? How is the cells? Now you will be wondering, the cell has an adenine, adenines over there, that is cadrins that are present over here, cadrins. Now this doesn't allow the cells to separate so easily. Now what is happening over here is FGF, fibroblast growth factor 8, is going to give signals to produce SNAIL, SNAIL, SNAIL1. That is again another type of a signal the cell will receive. Now, as long as the snail is present over there, the E-cadrins are tight. Now, the information is given to the snails to come out of the E-cadrin. So it is not anymore giving signals to E-cadrin. So what is happening is that when the E-cadrin is no longer attaching them, the cells get separated. As the cells get separated, they become loose and they start to move inside like this. Now, as they are moving inside, the first part, so you have this epiblast over here, you have this hypoblast over here. The hypoblast that is going to cover the yolk sac over there. Now, slowly, the hypoblast that is present over here is going to be displaced by the new cells that is going to be coming inside, and this forms the future endoderm cells over there. So you have this future endoderm cells above the hypoblast. And later on, after the hypoblast, the endoderm has been formed, there are more cells that are going to be coming inside by emboli. As more cells come inside over here, if you can see the cells that are coming inside. Now, these cells, the next cells are going to be formed, the future mesoderm cells. So the future mesodermal cells is going to be formed. Now, as the future mesodermal cells has formed completely over there, now the cells goes to the primitive node over there. Now that's a node over here. So if you can see over there, that's a primitive pit, and that's a Hence's node over there. Now there's a pit. Why you call it as a pit? There's a big hole over there. That's a pit over there. Now once, say for example, if I'm going to show them like this, now we have this groove over there. All the cells are going to be formed to form the mesoderm. Now the cells, the rest of the cells that are going to come and go inside this primitive pit, now this is going to form the future notochord. The notochord that is going to be formed is because of the cells that are coming inside, and they're going to form the future notochord. Now, once the notochord is formed, that gives the body access to the developing embryo. If notochord is not formed, then the body will not have a body access. So that is the importance of this primitive pit over there, where the cells from the epiblast is going to go inside and they're going to move forward anterior region. The notochord is not going to form to the caudal region. That's a caudal region, that's the anterior region over there. The cells that are going to move from this space over there, it's going to go forward over there. Now, this forward movement is called as a notochord. As the notochord is going to be formed, the primitive pit is going to become smaller in size. So here you can see the primitive pit is going to form smaller in size over there. So you can see, so this is what I was talking about over here, the primitive streak over there, how the cells comes inside. And as the cells are coming inside, they are going to replace the hypoblast to form the new endodermal layer. And the rest of the cells that are going to be formed coming over here inside is going to form the future, the mesoderm over there. As this is forming, now that is the mesoderm layer and the endoderm that has already been formed over there. Now that is the endoderm, the below one, that's the endoderm over there. That's the mesoderm over there. The mesoderm that is going to again split into two layers, the splanchnic and the somatic mesoderm. Now, as the cells are being formed, at the Hence's node, the cells which are going to be formed, that is going to form the notochord, and this notochord is going to move forward towards the animal pole. Now, as they are going to be forming into an animal pole, you can see the primitive streak is shortening in length over there. And as the primitive streak is shortening in length over there, now you can see the neural plate that has been formed over there at the anterior region. Why? Because the notochord has taken its shape. As a notochord has taken its shape over there, the mesoderm that has been formed over here, so the cells that are going to be forming the mesoderm is slowly going to give rise to somites. Now, what is the function of a somite over there? The somite is the one that is going to give rise to the muscles. So muscle cell is the one that is going to be formed by the mesodermal layer. So formation of the head process, that is a prospective notochordal cells over there. So that's a notochordal cells that are going to be conversion on the node. They sink inside. So that's a primitive groove. That's our Hence's node. They are going to shrink inside and they're going to form an elongated rod-like structure. So that is called as a notochordal rod-like structure that is going to be formed. And this notochordal is the one that is also going to give rise to some of the brain cells over there. What are the? It is going to form some glial cells over there. That's the, when you're talking about neurulation, we'll talk about that point in detail over there. And then, as I told you all, the primitive streak, it is also disappearing in length, and they're going to be very small, and they're going to be formed only at the tail end of the embryo. So that's a shrinking primitive streak over there. So a fully formed gastrula, what's happening to the fully formed gastrula over there when the primitive streak is completely disappeared? The fully formed gastrula consists of 3 germ layers. If you can see that 3 germ layers very clearly. Now, primitive streak has shortened up in length. So you have this rod-like structure, that's a notochord over there. The notochord is the one that is going to separate the embryo into left and the right, and then anterior, posterior regions over there. That is a body axis that it is going to give, and that's the head fold that is going to be present over here, the foregut that is going to be formed on the either side, and you can see the notochord in the center. Now, if you're going to take a transverse section over there and see the 3 germ layers over there, you have this ectoderm that is going to be formed. Then you have the neural tube in the center. And either side of the neural tube, you have the somites. The somites is again divided into 2 layers. That is the somatic layer and the splanchnic layer of the mesoderm. And below that, you have the endoderm, which is going to line the GI tracts over there. And of course, the dorsal aorta for the heart development over there. You have this dorsal aorta over there. So this is the point where gastrulation ends. So what is gastrulation over here? Gastrulation is formation of the three germ layers, that is the ectoderm, mesoderm, and the endoderm. So that was about the aves over there. And as I told you all, gastrulation in mammals is also as same as the chicken over there. So if you have understood gastrulation of the chicken, then mammal gastrulation is also easy. Now, what is the specific one that I have kept over here? So here you can see the ovaries over there, the ovary ovulating one egg, and the egg is taken by the fimbriae, the fallopian tube, that is a finger-like structures of the fallopian tube called the fimbriae. So they try to grab the ovulated egg, and once they grab it and put it inside the fallopian tube over there, if there is the sperm that is present in the fallopian tube, fertilization occurs. And once fertilization occurs, the zygote that is going to be formed is going to start the first cleavage over there. And the cleavage divides the cell into two cells, from two cells to four, four to eight, so on and so forth, forming a morula stage. Morula stage goes on to become the blastula stage, that is a blastocyst. And this blastocyst, when it has to go to the gastrulation stage, what happens is, now, at this place, when the egg has traveled into the uterus, the cells of the trophoblast layer that is present over there, mammal egg is made up of the trophoblast, outer trophoblast, and inner embryonic cells. The trophoblast is the one that is going to form a connection with the uterine wall of the mother's uterus. Why? Because that is the place where the placenta is going to be formed, and that is the place where the nourishment from the mother is taken directly to the child. And that is the reason why mammals are called as alecytal eggs. Why alecytal? There is no yolk at all. And a very little amount only during the development after the blastocyst and the gastrula stage. But later on, development has to, if the embryo has to grow, then definitely it has to have a connection with the uterine wall of the mother. Otherwise, the development stops over there. Okay, so here you can see how the development occurs, and we'll talk about gastrulation. As I told you all, gastrulation is the same as what we talked about in the aves over here. And so here, only difference between the aves and the mammals is aves development is outside, mammals development is inside the mother over there. So that is the mother is the one that is going to take care of the feeding part of the developing embryo. So the fetal organ involved in absorption of the matter and nutrient exchange of waste is taken care of by the placenta. The placenta, where the food or the nutrient from the mother is being given to the child by this placenta and the umbilical cord. Okay, so the placenta over here, what's happening to the placenta is, how is the placenta being formed? The placenta is being formed by the embryonic trophoblast cells. So the embryonic trophoblast cells is the one that is going to give rise to, so that's a trophoblast cells, and that is the one that is going to give rise to placental cells. Now you can see over here, so you have the inner cell mass, that is a trophoblast over there that is going to be formed. And that's the hypoblast, the epiblast and the hypoblast layers over there. And this is a trophoblast layer. The trophoblast layer is the one that is going to have a connection with the placenta, and they are going to form the connection by umbilical cord over there. How they are going to be formed is the one that we are going to talk about over here. So here, the epiblast delaminates into embryonic epiblast. So here you can see that is the epiblast over here. This epiblast is the one that is going to delaminate, and they're going to form the embryonic epiblast over there. And as this is going to be formed, the cavity in between these cells is called as the amniotic cavity. So here, so this cavity that is present over here, that's called as an amniotic cavity over here. And this amniotic cavity is the one that is going to take care of the developing embryo. That is the one that is going to act as a shock absorbers, the amniotic fluid that is going to be present in the amniotic cavity. Now, that is going to take care of the developing embryo and acts as a shock absorbers. And below that, you have this yolk sac, and the yolk sac is covered by this hypoblast layer over there. The yolk sac covered by this hypoblast layer over there, and also a little bit of trophoblast. Now, embryonic epiblast contains all the cells that generate the embryo. So this layer is the one that is going to generate the embryo. So how it is going to be forming over there. Now, if you can see over there, this blastula stage over here, the blastocyst cavity or the blastocele is present. Now, here I'm talking about after blastulation, the division of the cells that is after cleavage, whatever the cell you are going to get is a blastocyst stage. And you have this blastocele present over here. You have an outer covering called the trophoblast and inner cell mass called the embryoblast over there. So that's the embryoblast. And this is the cell that is very, very important for scientists. Why? Because these are the cells that you call it as totipotent cells. What is totipotent cells? Now, if I'm going to take out one cell from this embryonic cell and give them the growth factors, whatever the growth factors you want to grow them into, they're going to become that one. So that important those cells are. So they are something like magical cells. Whatever you want them to grow, they will start growing into that type of a cell you want. So that is called as the inner cell mass, or also called as embryoblast over there. Now, as they are moving inside the uterine, so that's the uterine wall over there, the endometrium, that is the uterus covering, is called as the endometrium. Now, this endometrium is the one that is going to get sloughed off during menstruation cycle. But during the zygote formation, a woman does not have menses. Why? Because the endometrium is still placed in the uterus wall over there. And this blastocyst, the one that is going to have a connection with this endometrium over there. Now, as they're going to close by, you can see in this picture over there, this the trophoblast that is going to cover them. Now, this is going to give rise to syncytioblast and cytotrophoblast. The trophoblast is going to form into two cells, syncytioblast and cytotrophoblast. Now, the syncytioblast is multinucleated. You can see those cells, there are many dots over there, and that dots are the nucleus over there. That's called as a multinucleated cells. And the cytotrophoblast is the one that is going to have mononucleation. So that's a mononucleation. Now, as the syncytioblast is going to grow, extend inside, now this is going to form connection with the blood vessels of the mother. Also, that's one blood vessel that is shown over here. Now, they are going to grow in such a way that they are going to have a connection with the blood vessels, so that the blood is the one that is going to transport oxygen and nutrients. So this blood is going to be in contact, the blood vessel is going to be in contact with the syncytioblast and the cytotrophoblast over there, which is going to form the future placenta over there. And this is the one that is going to supply the food for the developing embryo. So that's the embryo cells over there. This part, the blue color one, is the one that is going to form the embryo. Now, if you can see on the 7th or 8th day, a proper connection has occurred. So from this point, here it is only, if you say only 20% has gone in. Now here you can see almost 80% of contact with the endometrium has been done with the mother blood vessels and the embryo over there. And slowly an amniotic cavity is formed. And this is the one that is going to form the filled with amniotic fluid, and they're going to have connection, as I told you, shock absorbers over there. And that is the yolk sac that is going to be formed in the future. Now, just like in avian gastrulations, there is a place where a primitive streak is formed over there, as I told you all, in the chicken. There is one particular place called the collar from where the at the posterior side where the primitive streak is going to be formed. So something here also in mammals, it's going to be the same. So you have a primitive streak over there where the Hens's node or the primitive pit and the primitive groove that is going to be present. Now, as this is formed over here, that is the posterior part, you have this primitive streak that is going to be formed, and as the cells from the sidewards of the epiblast, they are going to go inside. Emboli is going to occur. As the emboli is going to occur over there, you have all these cells going inside into the primitive groove. They're going to come inside, and as they are moving inside, the hypoblast is going to get displaced and the new cells are going to be formed, that is going to be forming the future endoderm, and the rest of the cells that is going to be formed, they are going to be forming the mesoderm, the future mesoderm. And once the mesoderm is going to be formed over there, the whole cell that is going to be covered up, then the next what happens is goes to the primitive, the Hens's node or the primitive node over there. And the cells coming into this primitive node is going to move to the anterior region over there. And those cells are going to be forming the notochord. So I hope you have understood how this notochordal process is going to occur. So at the primitive groove or the primitive streak, the cells that are going to be formed, they're going to go inside and they're going to spread all over. And at the primitive groove, that's a procaudal region. Now, at the anterior region, there's something called a procaudal region over there that is predetermined. Now, the procaudal region is where the head is going to be formed. So, the notochord, once it is going to form, the cells that are going to go inside this Hens's node, they're going to form a rod-like structure, and they're going to keep moving until they touch this precaudal process over there. So, once they go up till there, the notochordal process stops over there. So that's a signal for them to stop over there. No more development, not more. The notochord is going to stop the development up till that place over there. And this place is called as a precaudal region. So I hope you have understood what is this primitive streak, the primitive groove. Streak is a streak. Primitive groove is a groove where the cell has to go inside, and you have a Hens's node or primitive pit over there, where the cells that are going to go into the primitive pit is going to form the notochordal process, that it is going to go near the anterior region to form the rod-like structure. Now, what is happening to this notochord in the future? Have you ever noticed? The cephalochordates, we saw the notochord still persistent. But is the notochord persistent in the mammals? No. As the development occurs, the notochord becomes nucleus pulposus. And where do you find this nucleus pulposus is when the vertebral column is formed. The vertebral column, so for example, if you have one bone over there and one bone over there, you have some vertebral columns like this, one below the other. That is the backbone over there. Now, between these two vertebral columns, there is something called a cushion-like structure that is going to be present. Now, that cushion-like structure is nothing but the notochord, the disintegrating notochord, and that forms a cushion-like structure, and the name of that is nucleus pulposus in the mammals. So here, I would like you all to again have a look about how the ingression of the cells occurs in the primitive streak over here. So you have this bilaminar layer of the is two layers. The upper one is the epiblast and the lower one is the hypoblast. Now that's a yolk sac over there. That's the amniotic fluid over there. So you have the cell that is covering amniotic fluid, and this is the cell that is going to be forming the future embryo over there for us. And that's a yolk sac. And as the cells are moving inside, you have the mesodermal layer and the primitive pit where the notochordal process is going to occur over there. And as I told you all, the trophoblast is the one that is going to give rise to the two layers over there. That is two layers. What are the two important blasts? Syncytioblast and the cytotrophoblast. Syncytio is the one that is going to have multinucleated. So these are syncytio layers that is multinucleated, and in between them you have the cytotrophoblast. And these are the one that is going to have connection with the mother's blood vessels, where the absorption of the food is going to take place. Now, as I told you all, this yolk sac is the one that is going to be in contact with this called the connecting stalk with the placenta. So this is the place where the placenta is going to form. And this yolk sac slowly changes into a connecting stalk over there, which slowly becomes the umbilical cord. And the umbilical cord is the place where the exchange of maternal food is going to occur from the mother to the child. And it also acts as the waste collecting region also, where the excess wastage is also being thrown out over there. Okay, so the trophoblast tissue and the mesodermal derived blood vessel constitutes the chorion, which fuses to form to the uterine wall to form the placenta. What is the placenta? Placenta is very, very important. Here you can see. So that is the placenta, finger-like structures that is going to be formed over there. And the placenta is the place where most of the exchange occurs over there. The placenta is the place again where, for example, if you want to have, say, for example, if you want to do some genetic studies from the child, the embryo, developing embryo, if you think that there's some genetic problem that is going to occur, you can also take out blood from the placenta or also from the amniotic fluid to do this genetic studies in them. Okay. So after this gastrulation, I would like you all, so this is about the gastrulation up till now, that is gastrulation. I have not talked about the development of embryo completely. What we did was, again, if you remember, we did the cleavage, fertilization, zygote formation, cleavage, blastula stage, gastrula stage. Now we are going to talk about neurulation. What is neurulation? How the head is going to be formed and how the heart also simultaneously. But here we are going to talk only about the head that is going to be formed over here. So that's called as neurulation. So what is a neurulation over there? So for example, neurulation in vertebrates results in the formation of neural tube. Now that's the neural tube. How is this neural tube going to be formed? Now, there's a information that has to be given to the the ectoderm above. So, if you can see over there, so this cell over there, now this cell, the anterior region is going to be given some information, which is going to be given the information. The ectodermal region is going to be given the information over there. So, the ectoderm over here, what happens is, the ectoderm over here is going to give information, that is the notochord is going to send signals to the ectoderm on above them to change its shape over there. As the change of the shape occurs, or as the information changes, they slowly change to form neural plate. Now, as a neural plate is going to be formed, they're going to form a bend around the notochord over there. As they're going to be forming on either side, there is one more information that is going to be given to the neural plate. That is, they are going to be forming the neural crest over there. Now, this is going to give information for the neural plate to bend over. As they are bending over the notochord over there, now you have this neural crest also being formed. And as they come close by, as they converge over there, the moment they converge over there, they're going to get cut off. The neural plate is going to get cut off to form a neural tube over there. And this green color one, which I told as a neural crest over there, that is going to form a sheet-like structure above the neural tube over there. Now, why am I stressing on the neural crest? Neural crest is also very, very important, as the picture says over here. They form the PNS, peripheral nervous system. So, peripheral nervous system, and also the neurons, the ganglions, the ganglia of the brain, the connections of the brain is all because of this neural crest that is going to be formed over here. And this neural tube that is formed, this is going to form the future brain. And this future brain is going to be in contact with extended form over there. And this extended form is the one that is going to form the spinal cord. Now, all this is because of the folding. And how the folding occurs over there? It's because of the epidermal layer that is going to form a plate. And then slowly, as a plate is formed, they're going to bend like that. And as they're going to bend like that, they're going to converge to form the neural tube over there. So this neural tube is the one that is going to be forming the future brain over there. And the neural crest, as I told you, is very, very important for different cells. It is also going to give rise to pigment cells also. The pigment cells of the dermis, the color is also because of this neural crest over there. And the notochord is necessary in order to induce neural plate formation. So the notochord is still there. Now, as this notochord, when does this notochord diminishes is when the vertebral column is formed during organogenesis over there. So here you can see neurulation, the somites formation. So this is how the bending occurs over there, the epidermal layer that is going to be bent over there. And as they are bending, they form a round neural tube over there. And as a neural tube is going to be formed, they're going to get chopped off. And beside them is the mesoderm that is going to be formed. And this mesoderm is going to again get divided into two layers over here, the somatic mesoderm and the splanchnic mesoderm over there. And here, this is a beautiful picture that you can see over here, how the development is occurring over here. That's a primitive streak, the brain plate over there, the neural plate formation that is going to occur. That is a neural plate that is going to have a bend and then they move upwards. So that is the movement of upward positions over there. And as the movement is occurring slowly, when the neural tube is going to be formed over there, as a neural tube is forming, the closing also occurs from anterior to posterior here. So in some animals, the closing of the neural tube is simultaneous, but in some, the closing of the neural tube occurs in the mid region and then next at the cephalic region and then at the caudal region over there. If you can see over here, the same thing is occurring over there. So you have a folding over there and slowly the head region is going to fold it and then the caudal region is going to get closed up over there. On the 23rd day, you can see the caudal region completely closing, and on the 22nd day, the anterior region is also going to get closed up over there. Now, this closure of the neural fold at the anterior and the posterior region is very, very important. Why? If the closure is not complete at the anterior region, then this ends up having an anencephalon. Anencephaly. Anencephaly is there is no formation of the head. And if the posterior region is not covered, then that leads to spina bifida. That is, the spinal cord is not well developed. At the posterior end, there is a big hole in the child, and that leads to spina bifida. We are not going to talk about the genetic problems of them. So in a nutshell, what all we did till now is we saw the blastomas forming the blastulation stage over there, the inner mast cell, the blastocele that undergoes the gastrulation stage, and the gastrulation stage gives rise to three layers: ectoderm, mesoderm, and endoderm. And after the formation, you also have this neurulation, where you have the neural plate formation, the ectoderm, endoderm, and the mesoderm that is going to be formed. So you have this neural tube, the mesoderm, the notochord over there. So here, what I'm going to talk about over here is during neurulation, this is of two types. There's one primary neurulation, and then you have the secondary neurulation that is good. Secondary neurulation that is going to be formed. Primary neurulation is how the neural tube is going to be formed and how the closing occurs. And in the secondary new relation, what is happening over here? The closing at the tailed region is occurring at the secondary new relation. So closing of the neural tube at the primary region and closing of the spinal cord at the secondary region is called the secondary new relation and primary new relation. Overview of what we did till now, the neurodevelopment over here, the gastrulation. Gastrulation is going to produce the new relation. New relation is going to mature to form the brain cells over there. So after that, we'll be talking about the limb development. Limb development will be done in the next class. So this was about the gastrulation, how the gastrulation is going to give rise to the germ layers, that is the ectoderm, the mesoderm, and the endoderm. So I hope you have understood a little bit of whatever we did over there. And still, if you have any doubts, you can again get back to us. And in the next class, we'll be talking about how the limb development is formed. So I hope you have understood this class, students. Sorry if there was some disturbance in between. See you all in the next class. Thank you.